Photosynthetic autotrophs are acquiring their energy by converting carbon dioxide from the atmosphere into the basic building block of organic matter, C6H12O6, otherwise known as glucose. So they're taking CO2 from the atmosphere, water from their roots, and then through a series of very complex reactions, converting that into our glucose molecule, some oxygen gas, and also releasing some water in the process. So where does photosynthesis happen? Let's take a look at the cross-section of a leaf. It is happening inside the mesophyll cells within the leaf. And then within those mesophyll cells, it's happening in the chloroplast organelles. Here we see a cross-section of a leaf. We see those mesophyll cells, and we see the chloroplasts inside those mesophyll cells. We also noticed that there is openings from the outside of the plant to the inside of the plant. These openings, or the stomata, allow for the exchange of gases that are required for photosynthesis. Photosynthesis, at a minimum, is a two-step process involving light reactions and then a light independent or dark reactions, also known as the Calvin cycle. The light reaction involves a series of complex and amazing steps that all get kicked off by a photon of light, specifically red and blue light wavelengths, which are referred to as photosynthetically active radiation, or PAR. This light raises the energy level of the photon receptors, which then kick off this hopping of electrons across receptors, much like the transfer of energy through an electric cable. Two outcomes of this electron transport chain that are critical to the production of glucose are the synthesis of ATP from ADP and NADPH from NADP+. These two molecules, ATP and NADPH, are required to fuel the dark cycle of photosynthesis, which is ultimately going to produce glucose molecules. The dark reaction, or the Calvin cycle, is where the inorganic carbon dioxide is going to become transformed into an organic molecule. This is referred to as carbon fixation. This is a cycle, so we could start anywhere, but we're going to start here with this existing 5-carbon organic molecule, ribulose biphosphate. This molecule is going to chemically react with the carbon dioxide, forming 2 molecules of a 3-carbon acid. This fixation of carbon dioxide, inorganic carbon dioxide, into an organic molecule is facilitated by the enzyme Robusco. This is pretty important because, ironically, Robusco is not very good at its job. It's not efficient at grabbing carbon dioxide from the atmosphere. So let's review the definition of an enzyme in case you need it. An enzyme is a biological molecule, protein, that accelerates or catalyzes a chemical reaction, acting as a carrier, connecting the molecules involved in that reaction. So Robusco is an enzyme that kind of leads a double life. Let's look at the full name of Robusco. It's ribulase 1,5-biphosphate carboxylase oxygenase. So the A's tells us it's an enzyme, and the carboxyl and the oxygen gives us an indication of what molecules it connect with. And this tells us that this enzyme Robusco works both with carbon dioxide and oxygen. And that's a little bit of a problem because to do this dark reaction, we need carbon dioxide. We don't need oxygen. We're looking at this here at the perspective of a leaf cross-section, and the green oval is representing a chloroplast, and the purple structure in the middle is representing a Robusco enzyme, obviously not drawn to scale. That Robusco enzyme has the ability to connect with oxygen or CO2 that is present in the internal structure of that plant. This is a problem because if it connects with the oxygen, it is not going to catalyze the formation of that 3-carbon acid, and then ultimately you're not going to get any glucose. However, it is a challenge, not a roadblock. For the majority of plants on the planet, it works. And Robusco has connected with CO2 and formed the formation of this 3-carbon acid. The next steps are highly dependent on that light reaction, using ATP and NADPH produced from that light reaction to form a 3-carbon sugar. This 3-carbon sugar, G3P, is then used to make glucose or potentially starches or other carbohydrates required for growth and maintenance. One really interesting thing too is some of this G3P does not get shuffled into any of those avenues, but stays in the Calvin cycle and becomes ribulase biphosphate, which takes us back to the beginning of the Calvin cycle. This describes the C3 photosynthetic pathway, the way that most plants on the planet do photosynthesis. In this process, carbon fixation creates a 3-carbon acid. The challenge with this pathway really comes back to this Robusco enzyme. In order to do this successfully, plants must have many stomata and keep them open often to ensure enough contact with carbon dioxide. Let's re-examine what's going on at the level of the cross-section of a plant leaf. So CO2 is entering inside that plant through the stomata. And the only reason that CO2 is going from the outside to the inside of that plant is due to diffusion. The concentration of carbon dioxide outside the plant is higher than it is inside the plant. And the reason that the concentration is remaining low inside the plant is that it is constantly being converted into that 3-carbon acid. So the internal CO2 concentration remains low due to its conversion into a 3-carbon acid. What is coming out while that stomata is open? Well, oxygen is coming out of that plant, but so is water. So water vapor is exiting out of the plant through the stomata. This is the process of transpiration, and it is also reflecting a diffusion process. The concentration of water is lower outside the plant than it is in the internal structure of that plant. So water is being taken up through the soil, through the vascular tissue of the plant, and ultimately out the leaf stomata to the atmosphere. This represents a trade-off to plants. Terrestrial plants must balance the intake of carbon dioxide, which they need to do photosynthesis, with the loss of water through the stomata. Now, stomata exist because the plants have to have that control to be able to close the gates. So plants can control water loss to some degree by the opening and closing of their stomata. Water use efficiency is going to be the ratio of the amount of carbon that is fixed, meaning inorganic to organic, carbon dioxide to glucose, via photosynthesis per unit of water lost via transpiration. This can be a really important constraint in arid climates. So it has driven terrestrial plant evolution and ecosystem productivity throughout the globe. So the environments where the C3 pathway is going to be most challenging to complete is going to be in those arid environments, those places where it's too costly for the plants to constantly have their stomata open to get enough CO2 because they're losing water and they can't afford that in those particular environments. Thinking back to how natural selection works, you have selecting agents, environmental conditions that favor some inherited trait over another, and targets of selection, those inherited traits that are expressing the pressure from the environment. In this case, the selective pressure is moisture availability. In these drier climates, plants using the C3 pathway are not going to be successful. In evolution terms, their water stress will limit their survival and their ability to reproduce and therefore their genes will be less present in subsequent generations. If there is any inheritable trait within the gene pool that reduces this stress, it will be favored and over multiple generations present in a significant portion of the gene pool. This process has led to the evolution of 3 different adaptations for how carbon is initially fixed from the atmosphere. These 3 different photosynthetic pathways are known as the C3, C4, and CAM pathways. The first thing to notice that's different about the C4 photosynthetic pathway is that it involves 2 cells. So in the C3 pathway, we only were working with the mesophyll cell. In the C4 pathway, we have the mesophyll cells, and then also the bundle sheath cells are involved in the process of photosynthesis. Second big difference to note is that CO2 is initially fixed into a 4 carbon acids in the C4 pathway. So we have this 3 carbon molecule called PEP combining with carbon dioxide to form a 4 carbon acid. This reaction is facilitated by a PEP coenzyme rather than Robusco. And the advantage of this is that the PEP coenzyme has a much higher affinity for carbon dioxide than Robusco. So it is more efficient at grabbing that CO2 and getting it inside the internal plant structure to allow it to facilitate this reaction. However, there are some drawbacks. It actually costs more to make the PEP coenzyme than it does to make Robusco. So there's some energy costs here. Once it's converted into a 4 carbon acid, it is diffused across into a bundle sheath cell, where it then breaks down back down into carbon dioxide. And you will notice that it then goes through the Calvin cycle. So it essentially is replicating the C3 pathway here. This step of carbon fixation, this initial carbon fixation, is a completely additional step in the process. Carbon dioxide is fixed by PEPCO, turned into a 4 carbon acid, hence the C4 pathway, and then is shuffled across to the bundle sheath cell, where it is going to break back down into carbon dioxide, undergo the Calvin cycle, and then eventually become a sugar. Notice that the Robusco enzyme is still going to be used here in the Calvin cycle. Once the CO2 is broken back down, CO2 will combine with ribulase biphosphate using the Robusco enzyme, making that 3 carbon acid, which then using ATP and ADPH will be eventually converted into a simple sugar. So why is the C4 pathway more water efficient than the C3 pathway? One, it's using that PEPCO enzyme that has a much higher affinity than carbon dioxide than Robusco. The stomatas do not need to stay open as long to get the same amount of carbon dioxide. Also, since the carbon dioxide is being immediately converted into a 4 carbon acid rather than a 3 carbon acid, the concentration of carbon dioxide inside the plant tissue is more quickly reduced. The numbers are lowered more quickly. So the concentration gradient from the inside of the plant to the outside is much steeper. And this causes quicker carbon dioxide diffusion. Remember, this is just happening. The movement of carbon dioxide from the outside atmosphere to the inside is just going by the process of diffusion, high to low. And converting it into a 4 carbon acid makes that internal concentration of CO2 even lower. So CO2 is going to come in even quicker. The end result of this, stomata do not need to stay open as long to get the same amount of CO2. And the quicker we can close the gates for having gotten enough CO2, the more water we're going to avoid losing. Clearly, C4 plants are more water efficient, and they're better able to capture carbon dioxide for photosynthesis. So why are they not more abundant? This map here shows us the percentage of native C4 dicot species in various regions of North America. Now, most C4 plants are grasses. We have sugarcane and corn are examples of C4 plants that use the C4 pathway to do photosynthesis. And you can see they're pretty small percentages throughout the US, and there does seem to be kind of some spatial patterns here of where we find higher percentages. So the reason more plants do not do the C4 pathway is due to the fact that it costs more energy. As we already have demonstrated, it costs more energy to make PEPCO. We're adding a whole additional step to the process, separating out initial carbon fixation and then undergoing the Calvin cycle and producing the sugars. So it requires 2 additional molecules of ATP for every molecule of carbon dioxide produced. In addition, it's not very efficient at low light intensities, so the C4 pathway does not work very well in shade. Obviously wouldn't work very well in temperate deciduous forest biome in which we live. And finally, it requires a higher temperature optimum for growth. C4 plants are going to do better in warmer climates. We can kind of see that here with the percentage jumps up a little bit in the southwest United States. The CAM pathway has evolved in the most water-limited environments on the planet, and it has some similarities both with the C3 and the C4 pathway. The biggest defining characteristic of the CAM pathway is we have these diurnal patterns in which the stomata is closing during the day when it's very hot and you can have high rates of evapotranspiration, and then they open during the night. So what's happening is carbon fixation is occurring at night when the gates of the stomata can open up because the risk of transpiration water loss is lower. So we're going to open up the gates to get the CO2 in, and that's the time of the day when they're going to be at a minimum of water loss coming outside of the stomata because temperatures are much lower. So like the C4 pathway, that initial carbon dioxide fixation is going to use a PEPCO enzyme and create a 4-carbon acid, just like the C4 pathway. These 4 carbon acids are then stored in vacuoles in the mesophyll cells until daylight. Now, this is one way that the CAM pathway is similar to the C3 pathway in that the whole reaction is happening in the mesophyll cells, unlike the C4 pathway where it's happening in the mesophyll cells and then the 4-carbon acid is being shuffled across to the bundle sheath cell. Here it's all happening in the mesophyll, but it's being stored in these vacuoles until daylight. And then when daylight happens, the gates close because we don't need the stomata open to get light. So the light is going to hit the photon receptors, cause that electron transport chain to happen. We're going to produce ATP and NADPH, which is needed for the Calvin cycle. So our 4-carbon acid that's been stored in the mesophyll is then released. It's broken down into carbon dioxide, combines with ribulose biphosphate, using the Robusco enzyme to create PGA, which eventually will become our glucose molecule. This is the Calvin cycle. In summary, all 3 photosynthetic pathways involve the light reaction, which produces ADP and NADPH, and the dark reaction, also known as the Calvin cycle, which converts CO2 into a simple sugar using that ATP and NADPH produced in the light cycle. The difference between these pathways is that the C4 and the CAM pathway have an additional step for that initial carbon fixation. This additional step allows these plants to be more water efficient. So in a C3 plant, that Robusco enzyme is the one that initially fixes carbon dioxide and directly takes that into the Calvin cycle to produce a sugar. In the C4 and the CAM pathway, the enzyme that is initially used to fix the carbon dioxide is the PEPCO enzyme. In the C4 pathway, that'll then get shuffled across to a bundle sheath cell where it breaks back down to carbon dioxide and goes through the Calvin cycle. In the CAM pathway, everything happens in the mesophyll cells, but the C4 acid that's created is stored in these large vacuoles in the mesophyll cells until daytime. So the final difference here with the CAM pathway is that it's doing that initial carbon fixation at night. And it's going to store that carbon acid in those vacuoles until daytime when the stomata close. And even with the stomata closed, the photosynthetically active radiation is going to stimulate the electron transport chain to produce ATP and NADPH required for the Calvin cycle to convert the carbon dioxide into a simple sugar as it does in all the other pathways. The other key differences here is to recognize the optimum temperatures on which these pathways are going to be best. So highest temperatures, the CAM pathway is going to be most efficient. And at the medium temperatures, we have the C4. At the cooler temperatures, the C3 pathway is going to be most efficient. Water use efficiency, remember that how many units of carbon dioxide can produce per water unit. And the water use efficiency is going to be best for a CAM plant and least efficient for the C3 plant. But remember, the most energy efficient pathway is going to be the C3 pathway, which is why most plants are C3 plants, and we only see the percentage of C4 and CAM pathways as more prevalent in these drier climates.